Our next speaker is Dr. Michael Taylor, who will be speaking to us about intra- and intertumoral heterogeneity in medulloblastomas that introduce barriers into effective treatments.
Thank you. I was initially going to thank Dr. Rao for inviting me to share some of our work today, and then I realized he gave me a talk right before someone who says talent is overrated, so I haven't made my mind up if I'm going to thank you or not yet, Ganesh. So my lab focuses on medulloblastoma, and probably everybody here in the crowd has taken care of these patients or operated on them at least once in their career, so I'm not going to give you a big introduction. We've known for some time now that medulloblastoma has molecular heterogeneity. So we're probably not dealing with just one homogeneous disease, but rather a number of diseases. And there are a number of papers over a number of years here where we showed that at a molecular level, there was more than one disease, including one from my own lab. We had a problem, though, that we really couldn't resolve the barriers between the different types of medulloblastoma because nobody had enough patients. And then when I returned back to Toronto to take my staff job, I'd managed to collect up about 100 medulloblastomas. Then we studied them by transcriptional profiling, which is measuring the level of expression of every gene in the genome. And then we asked the computers, which one of these things are most like each other? And when we did that, we got four very crisp, clean groups of medulloblastoma. The so-called Wnt group, because it has overactivity of the Wnt signaling pathway. The sonic hedgehog group, because it has overactivity of the sonic hedgehog signaling pathway. And two less descript, previously called group 3 and group 4, or C and D. These four groups have different demographics, different epidemiology, different transcriptomics, and different genetics. There is no such thing as medulloblastoma. There are four different diseases. Medulloblastoma is Bigfoot. All right, so then when we went on to classify medulloblastoma, and we were doing this, we were using these microarrays, right? Microarrays are expensive, they're fussy. If you went home and told your pathologist you wanted him to do microarrays, he'd probably have a stroke. Right? He'd have to be stented by the guys in Buffalo. So rather, we need a test that we could take that the pathologist will actually like. And so what we did is we looked at the genes that were very highly overexpressed in each of the classes of medulloblastoma, as shown by the red level of expression there. And then with the help of my friend Stefan Pfister in Heidelberg at the DKFZ, we identified antibodies to the proteins encoded by those genes. And what you can see now is that we can stain the tumors and identify by immunohistochemistry which tumor belongs to which subgroup. And I'm not a pathologist, but it's, brown, not brown, brown, not brown, brown, not brown. So now we have an immunohistochemical test so that we can tell the difference between these four groups. So does this really matter? Who cares, Michael? You're in your lab spending all this time. I'm sure you're publishing papers and padding your CV. Well, it probably actually does matter and matters on a clinical level, because here's the outcome for the four different groups of medulloblastoma. If you have a Wnt tumor, shown by the blue line here, you are going to survive. The survival rate for Wnt tumors is almost 100%, and the patients that die are dying of the therapy or of secondary glioblastomas that are caused by the therapy. If you have a group 3, here labeled group C tumor, you are going to die, almost certainly. And that's even in the face of modern therapies that include high-dose chemotherapy with bone marrow transplant and craniospinal radiation. So these guys we're probably giving futile therapy to. And if you think about it, if the survival rate here is 100%, we're wrecking these kids, giving them craniospinal therapy. We can probably improve their outcomes just by walking over to the radiation machine and turning the dial down. In the past, the most important prognostic factor in medulloblastoma was whether or not you had metastases, so cells that had spread through the leptomeninges. That was the #1 risk factor. In fact, though, once you account for the subgroup of medulloblastoma, Mets don't matter anymore. If you've got a metastatic Wnt tumor, you still survive. It's not significant, but you actually do better than a localized Wnt tumor. If you've got a localized group 3 tumor, you still die. And in fact, metastases are much more common in the group 3 tumors, and when metastasis was a risk factor in the past, it was probably just a surrogate marker for group 3. So here we have this small blue cell tumor. It's got some evidence of rosette formation. Is this, we're talking to the parents now, is this a Wnt tumor which their child is almost certainly going to survive? Or is this a group 3 tumor where their child's going to suffer horribly and then die? So we know that medulloblastoma is 4 distinct diseases. We can identify which of the 4 diseases they are by using immunohistochemistry, another technique I won't talk about today called nanostring. In my laboratory, if you ship us flash frozen tissue, is willing to do this for free for anyone in the world. We could do it on about a 2 or 3 week turnaround to determine the subgroup. Because the genetic events vary between the diseases, the targeted therapies are going to need to be different. Families are already starting to ask their surgeons and their oncologists around the world what their subgroup is. Most of our data on the role of gross total versus subtotal resection in medulloblastoma is based on unsubgrouped tumors. So we probably need to look at that again. So to agree on the nomenclature for the molecular classification of medulloblastoma, if there is such a thing, we had an international consensus conference about a year or two ago. And a consensus is defined as something that nobody likes but everybody can live with. And we eventually came to this classification of Wnt, sonic hedgehog, group 3 and group 4 medulloblastoma, and we published this consensus paper. And it's freely available on the internet, as is this figure that describes the 4 subgroups. So now we come back to this. Is this a Wnt medulloblastoma or a group 3 medulloblastoma? Actually, it's neither. It's the driveway of this very nice chateau in Burgundy that I saw riding past on my bicycle. So it's really just gravel. And I like to convince you that 5 years from now or so, if you're classifying your pediatric brain tumors just on the basis of histology, that you're really stuck in the Stone Age. So again, Michael, how does any of this really matter? So remember I said that one of the subgroups is called the sonic hedgehog subgroup. There are drugs that target the sonic hedgehog pathway. And this paper published by Genentech in the New England Journal, they took a patient who had widely metastatic medulloblastoma, gave him the sonic hedgehog blocker, and the tumor went away. It did come back later, and more clinical trials are ongoing, and the data's not out there, but there's a very high response rates to these drugs, and some of them are durable. Surgically, some of this stuff is very relevant as well. When we all operate on medulloblastomas, we often talk about a medulloblastoma that has invaded the floor of the fourth ventricle. The assumption always being that the tumor started in the cerebellum and secondarily chewed its way into the medulla. In fact, there's some nice evidence now from Richard Gilbertson's group at St. Jude showing that for the Wnt tumors in particular, they actually start in the medulla and then invade the cerebellum. And the irony here is these are not the ones that you want to chase and cause a horrible deficit on, because these are the kids who do so well. So to understand the genetic basis of medulloblastoma, because there is this intertumoral heterogeneity, when I came back to Toronto, I formed something called the Medulloblastoma Advanced Genomics International Consortium. And the slide says 45 centers, but it's actually out of date. We now have over 60 centers, 60 cities around the world who have joined MAGIC and sent me flash frozen medullos in Toronto. And we have over 1,500 frozen medulloblastomas to look at. Since we're looking at 4 diseases, now we're talking about 300 or 400 from each group, and maybe we have the power to actually look at them. So we got a $10 million grant from Genome Canada. And the first step that we did is we looked at 1,200 medulloblastomas using Affymetrix SNP arrays to determine copy number, so to look for amplifications and deletions in the medulloblastoma genome. And what we found is that most of the amplifications and deletions in medulloblastoma are indeed subgroup specific. Wnts are different from hedgehogs, and hedgehogs are different from 3, and 3s are different from 4s. We did find, luckily in a few cases, events that are highly targetable, for which there's drugs on the shelf that are used for other cancers or non-neoplastic conditions that we can repurpose for medulloblastoma, such as the TGF-beta pathway that's overactive in these group 3 medulloblastomas. If you remember, these are the kids who all die. And we can see that the TGF-beta pathway is activated in about 20% of kids with group 3. There's no reason we can't take those drugs now and test them in a phase 2. Previously, no fusion genes had been known in medulloblastoma. They're very common in things like leukemia and some sarcomas of childhood. What we found when we were doing some RNA-seq, which is sequencing the entire transcriptome of medulloblastoma, was recurrent fusions between a non-coding gene called PVT1 and the gene called MYC, which is an oncogene that probably even the spine surgeons have heard of, and that these 2 genes were in fact fused together to form a fusion gene. And what this results in is normally MYC's promoter is downregulated by MYC, but instead now you have the MYC gene downstream of the PVT1 promoter that's turned on by MYC. So you have the MYC gene making MYC, the MYC gene then goes around, turns on the PVT1 promoter, and it makes MYC, and it goes in a vicious cycle. No wonder that causes cancer. The most common region of amplification in the medulloblastoma genome was a very focal area on chromosome 5. It was only found in group 4 tumors, and it affects a single gene called SYNCAP. SYNCAP may be familiar to some of the functional surgeons here because when it's mutated in the germline, it causes Parkinson's disease. You don't have enough SYNCAP, your dopaminergic neurons die, and you get Parkinson's disease. However, if you have amplification of SYNCAP, which we went on to prove by whole genome large insert sequencing, you have too much SYNCAP around, the neurons that are supposed to die, so some of the progenitor cells in your cerebellum don't die, and you get medulloblastoma. It's like Goldilocks and the Three Bears. Too much SYNCAP, medulloblastoma. Too little SYNCAP, Parkinson's disease. We looked at over 4,000 human cancers, and we only found duplications of SYNCAP in group 4 medulloblastoma, showing just how specific it is. So we looked at 1,000 medulloblastomas here, over 1,000, and too much was almost enough. The targets for therapy in medulloblastoma at a copy number level are very subgroup specific. For some of these targets, there's drugs already on the shelves that we could test at the level of a phase 2 trial. Medulloblastomas are a copy number driven disease, by which I mean they have very few somatic mutations or single nucleotide variants, as we published in a couple of papers in August, whereas they have lots of copy number things. And some of these actionable events should transition soon to clinical trials. So there's about 6,000 papers in the literature on medulloblastoma if you go to PubMed. And probably out of those 6,000, all but about 5 are really primarily focused on primary medulloblastoma. But as neurosurgeons, that should be kind of stupid to us. In fact, you guys should probably be pelting me with rotten tomatoes right now, because as neurosurgeons, we can cut out the primary tumor and throw it in a bucket. There is not a neurosurgeon in this crowd, or who has ever lived, who can remove all of this leptomeningeal disease from this child and have them come out in a functional manner. Beyond that, the horrible treatments that we give, like craniospinal radiotherapy, are really aimed at this leptomeningeal disease. So why are we all studying the primary tumor and not the Mets? So in fact, we've had this assumption over time that the primary tumor and the metastases are the same. But when I talk to my oncologists, that's not actually true. Even in the face of non-specific cytotoxic therapy, you can see here's primary tumor and metastasis. We give chemotherapy. All of the Mets are gone, not some of them, but all, but the primary is untouched. And conversely, in this patient, primary tumor and metastasis, we give chemotherapy. The primary is all gone, but the metastases are untouched. So there's some evidence at a clinical level that medulloblastoma metastases are different from the primary tumor. My laboratory made a new mouse model of medulloblastoma using a functional genomic technique called Sleeping Beauty, where this transposon jumps around the genome, randomly turning genes on and off. And not only did we get primary medulloblastoma, as you can see here, but in fact the medulloblastoma is metastasized and coated the spinal cord and coated the brain. We then use Illumina next-generation deep sequencing to look at the mutations in the primary tumor and the Mets in these two different mouse models. And we were shocked. We were shocked at first because the overlap between the primary and the Mets was only about 10%. They were the same tumor for sure, but the assumption that things you discover in the primary tumor are going to be present in the Mets was completely and totally false. Around this point in my talk, somebody usually says to me, well, Michael, mice aren't children. So we went on to look at now some human pairs where we had human medulloblastoma metastases in primary tumor. And just showing very nicely here, here's MYC amplified in the primary tumor, not in the Mets. MYC amplified in the primary tumor, not in the Mets. Because FISH is sort of a 1970s technology, we then went on to do whole genome sequencing on a number of these samples where we looked at the primary tumor and the metastases. So this is sequencing all 3 billion base pairs of the genome at 30 times coverage using Illumina. And what we can see is only about 1/3 of the mutations in any given medulloblastoma are shared between the Met and the primary. And most of the mutations, or at least a preponderance of the mutations in the Met, are not in the primary tumor. So if we go around studying the primary tumor and try to give drugs to treat the metastasis, we're not being very bright. When we looked at cases where there was more than one metastasis, either by CpG methylation, by copy number events, or by sequencing and looking for somatic mutations, what we could see is multiple Mets from a single animal or from a single child were very similar to each other. So really, if you have medulloblastoma, you have two diseases. You've got the primary disease and the metastatic disease. So we're calling it bicompartmental disease, or bicompartmental medulloblastoma. So the Mets are distinct from the primary. The metastatic medulloblastoma is a bicompartmental disease. And we're going to need to start biopsying these as neurosurgeons once targeted therapies are available so that we can help guide targeted therapy. One of the other big assumptions that we make is that we all study, and by we I mean those of us who study medulloblastoma in the lab, study untreated primary medulloblastoma. We go to our lab and spend millions of dollars and even more hours. And then we turn around and identify drugs, and we use the drugs on children who have highly treated disease after radiation and chemotherapy because we assume that the targets will still be there. We assume that. In fact, all of the trials that are going on at COG are based on that right now. And we're putting all of our patients through these trials based on that. So we started to develop a mouse model using our leptomeningeal disseminated mouse model that I showed you before. And my Italian postdoc, Livia Garzia, developed the skills to operate on mouse medulloblastoma by putting the mice to sleep. She initially did this with her bare eyes, and recently Jim Rucka gave her a pair of his old loupes. And she can do a subtotal resection on mouse medulloblastoma and have the mice survive. After that, the mice recover, and they go for multifractionated CT-guided craniospinal radiotherapy, the same way our children do, over a period of about 3 or 4 weeks. You can see there's our collimators, and they're getting radiation along their spine into their entire cranial compartment. And when she does that, and don't worry about all the data on here, when we sequence the primary tumor that she took out at surgery versus the tumors at the time of recurrence, we can see that they're extremely different. The recurrence comes from the primary tumor, but from a very limited subclone, and a lot of the targets that were there in the primary tumor are no longer there at the time of recurrence. One of the top genes that's recurrently mutated in the recurrent tumors is P53, the most famous tumor suppressor gene of all time. So again, now, Michael, this is a mouse model. It's not a human, so we went back and we managed to collect a number of human samples where we had tumor taken before any therapy from a human child with medullo, and then tumor taken at the time of recurrence. And these are mostly recurrences in the posterior fossa as opposed to metastatic recurrences. And here, just looking, this is an untreated primary tumor, and you can see all these copy number gains and losses as we go up and above there. And you can see that in the recurrence, most of the events that were in the primary are lost, and then there's new events. So how well do you think this strategy that we're using at COG is going to work if we keep doing this? So that was just at a copy number level. We were also doing this now at a level of whole genome sequencing, again using Illumina technology. What we can see is there were 236 mutations in the primary tumor that weren't in the recurrence, and 290 in the recurrence that weren't in the primary tumor. So that assumption was wrong. And if we keep going down this pathway, we will never make any progress at COG. We're just going to keep running phase 2 trials until the end of time. Medulloblastomas, when they recur, they do stay in the same subgroup. As opposed to glioblastomas that can start out as a good subgroup and become a bad subgroup after therapy, whenever we look pre and post therapy, the medulloblastomas always stay in the same subgroup, but the mutations are different. We noticed a very important pattern with regards to recurrence as well, and I think this is surgically important. When we look at the sonic hedgehog medulloblastomas that recur, they almost always recur locally, not metastatically, as opposed to the group 3 and 4 tumors that almost always recur metastatically. So I think there'll be a larger role for reoperation locally in the posterior fossa in the sonic hedgehog tumors. So in conclusion about primary recurrences, recurrent tumors are extremely different from their matched primary tumor. The targets that we identify at diagnosis aren't present at recurrence. So if we identify something like a notch amplification in the primary tumor, and then assume it's there at recurrence and give the drug and the drug doesn't work, it isn't because the drug didn't hit the target, it's because the target's not there anymore. And we need to redesign the way we're doing clinical trials. There's also new targets in the recurrence. For example, we keep seeing things in the hedgehog tumors that are hitting the insulin-like growth factor pathway, which is eminently targetable. And we're missing that by not looking at the right compartment. I think biopsy of recurrent medulloblastoma is going to become much more common with the availability of targeted agents. So along with biopsying the mets, I think we're going to have to biopsy the recurrences. And these aren't really the operations we all like to do. We all like to resect big tumors. We don't like to go in there and chisel out a little piece for the oncologist. But I think this will make up an increasing part of our workload as pediatric neurosurgeons as we move into the next 5 to 10 years. So there's no such thing as medulloblastoma. Medulloblastoma is Bigfoot. There's 4 different diseases. The subgroup affiliation is clinically and surgically relevant, and we'll do it for you free of charge. You have to pay for the shipping. The metastases in the recurrent tumors are divergent and likely require biopsy in the near future as targeted agents come to the clinic. Targeted therapies are actually already here, as I showed you with the hedgehog inhibitors. Those are some of my funders, and that's my lab and my many collaborators. Thank you very much.